We are now privileged to hear from Dr. Marianna Rossetti, who will guide us through the exciting field of DNA nanotechnology for bioanalytical applications. Dr. Marianna Rossetti is a senior postdoctoral researcher at Catalan Institute of Nanoscience and Nanotechnology, specializing in DNA nanotechnology for diagnostics, imaging, and drug delivery. With a PhD from the University of Rome for Vergata, her interdisciplinary research integrates analytical chemistry and material science to advance point-of-care diagnostics, including application for Alzheimer's disease. Dr. Rossetti, thank you for joining us. So please take the stage.
So I hope that you can hear me without the phone, because I know that in this country you are really tall, all of us. It's really complicated. Okay, but okay. First of all, thanks for the organization and to inviting me. And in this presentation, I will try to give you a brief overview how can we use DNA nanotechnology for a bioanalytical application. And but first of all, like Claudio, I just want to present myself briefly. And I am a chemist. I got my bachelor and master degree at University of Rome La Sapienza. After that, I moved to the University of Rome Tor Vergata, where I got my PhD in chemistry with a thesis on DNA nanoswitches for bioanalytical application. After that, I was granted with 3 different fellowships from Fondazione Umberto Veronesi, that is an Italian foundation that found research on cancer and also an application for the detection of cancer disease. And so for 3 different years, I worked in Roma Tor Vergata in order to develop sensor based on the use of DNA for the detection of breast cancer biomarkers. After that, I won a Marie Curie individual fellowship that bring me to the Catalan Institute of Nanoscience and Nanotechnology in the group of Arben Mercoci, with the idea to combine my expertise in DNA nanotechnology with the expertise of the group, in particular by combining DNA nanotechnology with nanomaterial, in particular with 2D material. My fellowship ended at the end of January 2022, and now last year. I started in 2022, and now I'm working in the same group in the frame of 2D Biopath. That is an European founding project. Then I will show you briefly at the end of this presentation. During my PhD, I also spent a visiting period in Canada at University of Montreal in the group of Alexis Vallée-Belize, where I developed a DNA-based sensor for the detection of urea in whole blood. And this brings to the creation of a startup, and then a patent is on the way to be released. And in 2018, I had the opportunity to spend some months in the group of Michael Saylor at University of San Diego, and where I had the opportunity to okay. So I was, okay. Okay. I was in the University of San Diego, where I had the opportunity to attend this summer school for silicon nanotechnology with the aim to combine DNA nanotechnology with silicon material. And then to conclude, I work in the group of Nora Ventosa, where I combine DNA nanotechnology with the lipid-based material. So that's it for saying that in all my career in these 10 years of DNA nanotechnology, I try to combine DNA nanotechnology with different kind of material. And it is what I want also to highlight with this slide. Then I had the opportunity to spend different visiting abroad, because I had the opportunity to work in different European projects. That is, this at the beginning was called IRSIS, then now the name is RISE, that are projects in which different partners write a project in order to have a staff exchange. This is really useful because give us the opportunity to move and also with a good salary. That is important, in particular when you are a PhD and to move away, in particular, if you are not doing the PhD in a country that is your, in a city that is your city, you have to also pay your flat in one country and another one, so the salary is could be an important factor in the decision. Okay, but moving to the science and in what we want to try to do is to mimic nature. Because we, in particular, to mimic the structure switching biomolecules, because we know that in nature there are many kinds of bioreceptors that work with a conformational change. What does it mean? That there are some bioreceptors that are able to recognize their specific target, and after the recognition event binding, they can change conformation. And many times this conformation brings to a secondary activity. For example, the opening of an ion channel, for example. But why we want to mimic this kind of bioreceptor to do biosensing? Because we know that these mechanisms are really specific. In fact, with the nature use this mechanism because they are really specific and they are able to work in a really complex environment like the cellular one. So to mimic this kind of mechanism give us the opportunity to create biosensors that are high specific and high sensitive. Which kind of material we can use to create this kind of DNA nanoswitches? In one of the materials that is really useful to do that is the DNA. When we talk about DNA nanotechnology, we refer to the field of science in which the DNA is used like a material and not like carrier of genetic information. So we are talking about synthetic DNA. But why to use DNA and not another material, for example, protein material, amino acid or like this? Because the DNA is today is a material that is really easy to synthesize, and this means that also the cost is quite low, and also because the chemistry is quite easy, so that it is really easy to modify with a wide range of molecules, for example, redox tag, optical tag, or other kind of biomolecules, or a peptide, for example, and also because this material is easy to buy. So also if we have not expertise in chemistry, also is could be a click chemistry or not really complicated chemistry, we have the opportunity, like Claudio showed before, to buy from different companies. And why the other important thing is that the nice thing of the DNA is that it's easy to predict the structure of a DNA sequence, because in this case, we are talking about DNA sequences that are not really long, so that we can know a priori what is the structure of the DNA sequence can adopt. And in fact, we know that adenine can bind just thymine and guanine can just bind cytosine. And we can also to use different software that are free online, for example, NUBAC, like Claudio showed us, in which we can we can write our DNA sequence, the primary structure, and then we can also, for example, to add different strands. And then, thanks to this software, we can see not only the structure, but also the interaction between different strands of DNA in order to see a priori what is the binding, if the structure is useful or not for our application. Okay. But when we talk about DNA nanoswitches, we have to think, the first thing is to think at what is the physics of this kind of bioreceptor. Okay, in this kind of bioreceptor, we want to mimic what happens in nature. So that means that we want a bioreceptor that can exist in two conformation, a non-binding state and a binding competent state, that are in equilibrium with each other, with a constant the constant of switches that regulate this mechanism. Okay, the target can bind just the bioreceptor when it is in the binding competent state. So when we have this conformation, we have the binding, the formation of the target receptor complex. Okay, so when we create a sensor, what does it mean? That what we are looking is not the real affinity of the bioreceptor and the target, but is that we call a apparent KD. And because we have to take in account also the constant of switching. So when we design a DNA nanoswitch, so a nanoswitch in general, we have to think that this constant is that give us the affinity that we see. This means that, for example, if we design this is regulated by this equation, in which we can see that if, for example, we design this switching in order that this is too stable, for example, and then, for example, we have that we have a population, for example, of 100 molecules, and we have 90 in this conformation and just 10 in this conformation. What does it mean? That the KS in this case is 0.1. This means that our binding curve will shift toward right. But if we, for example, design the nanoswitches in order to have half of the population that is in the non-binding state and the other half population in the binding competent state, we will have that the half of the target will bind the competent state and not the other one. So what is important that when we have also this binding, we have that this equilibrium is shifted toward right for the equilibrium, for the thermodynamic equilibrium. So if we design in general a switch that have a constant switch in between 0.1 and 1, we have the good compromise for having a good signal, but also a good affinity. In fact, if we design a switch that has a KS of 1, that means to have half of our receptor in non-binding state, the half bioreceptor in the binding competent state, we will have that we will have a signal that could be the 50%, okay? But this means that our affinity is decreased just to order of magnitude of the real affinity, because in this equation we will have KD is 1 plus 1 divided 1. So how can we design the easiest DNA nanoswitches? The easiest way to design a DNA nanoswitch is what is called molecular beacon. The molecular beacon is a single-strand DNA with the two extremities are self-complementary in order that this molecule can adopt this stem loop structure, where the stem is composed of the two self-complementary sequence at the extremity and the loop. Okay. This stem loop structure is in equilibrium with an open structure. Okay, so if we design this loop in order to be complementary to the DNA sequence or RNA sequence that we want to detect, what we will have is that in the presence of the target will bind the loop, moving this equilibrium toward right. Okay, but now how can we see this? Okay, the easiest way is in fluorescence. For example, if we modify the two extremity with the fluorophore and quencher pair, we can have that in the absence of the target, we will have this stem loop conformation in which the fluorophore will be close to the quencher and will have a low fluorescence signal. But in the presence of the target, we will have the opening of the stem structure. So that means that the fluorophore will be far away from the quencher and we will have a high fluorescence signal. So in this case, we can do the association between the increases of the fluorescence with the concentration of the target. To move this from the fluorescence to electrochemical detection, we can modify our molecular beacon with the thiol group to one extremity in order to attach, for example, on the cold electrode, and modify the other extremity with the methylene blue. So as Claudio showed before, in the absence of the target, we will have that the methylene blue will be close to the electrode surface. That means that we will have a high electron transfer between the redox stack and the electrode surface, and so we will have a high current signal. When we have the target instead, the target will bind the loop of the molecular beacon. We will have the opening of this stem loop structure. That means that the methylene blue will be far away from the electron surface. So that means that the electron transfer is lower. So we will have a decrease of the electron current. But how can we use this kind of a molecular beacon to detect targets that are not oligonucleotide, okay? We can use the DNA like a template. That means that in this case, the DNA is not the recognition element, but the DNA is just the template that allow to have a different signal change. In this case, for example, we modify the molecular beacon with two tails at the extremity. These two tails can hybridize with a second strand of DNA that present recognition element. Okay, this is really useful, for example, for the detection of antibody. Why? Because the antibody have the characteristic to have the two different epitopes that are distant more or less between 10, 15 nanometer. So that means that if we design this strand and modify with the specific antigen, we will have that the antigen will bind the first site, and then binding the second side will open the stem loop structure, because this distance is 10, 15 nanometer. That means that at the beginning we will have a low fluorescence signal, okay, because the fluorophore will be close to the quencher. But when we have the target will open this stem loop structure and we'll have the increase of the fluorescence. What is also the peculiarity of this kind of biosensor is that this is really modular, because by just changing the recognition element, we can detect different kind of target. This is really useful when we develop a platform, because in this way, by just changing a small part of the sensor platform, we have the possibility to detect a wide range of analyte. And for example, we did this modifying this strand, for example, with DMP, that is small molecule, with peptide, for example, the P17, and with the aptamers, in order to have the specific detection of anti-DMP, okay, in this case by modifying this tail with DNP, or to detect the anti-HIV-AB by modifying this DNA with a P17 peptide, or by adding aptamer, for example, in the case that we tested was for the PDGF detection, we can modify this strand of DNA with a PDGF aptamer in order to detect PDGF. And we can see that we have a really good result in terms of specificity also, and also signal gain. But moving in a little bit more in advanced idea, when we talk about antibody, one of the really nice thing that I tried to explain already is that they are the specific characteristic that they have a distance between the two recognition, two epitope that is more or less 10 or 15 nanometer, more or less. And so that means that this characteristic can be used to exploit co-localization effect. That means that, for example, if we have two DNA sequence, okay, the same that we have seen before, that are modified with the recognition element, if they are free in solution, they cannot bind if the affinity between the two strands is low. But if we have a template that bring these two strands in close proximity, this means that we have an increase of the local concentration of the two strands. So that means that these two DNA strands, also if they have low affinity, they can bind. They can bind because we have like a really high concentration. It's not more nanomolar concentration, but it's like to have a micromolar concentration, depending on the distance in which we constrain to stay the two DNA strands. And this kind of mechanism has been widely exploited in the last year for different kind of application. For example, biosensing, also chemical synthesis, chemical reaction, because we can put together two reagents in order to do the same reaction that we have to need, that we have in other terms we have to do in higher concentration, in lower concentration. So this is really useful. Also in catalysis. We use this principle of the colocalization effect in also another times to detect antibody. This time moving to an electrochemical platform. So what is the idea is repeating is that if we have, imagine to have two DNA strands, one that we call capture strand, that is a strand that is attached on the electrode surface, and the second strand of DNA, this we can, in the cartoon is in orange, and then we can modify with the redox tag like methylene blue, and that we can call output strand. So these two strands has a low affinity. So that means that in order to have the binding between the two, imagine that this capture strand is fixed because it is mobilized. If we do a binding curve with the up strand, we will need a high concentration in order to have the binding, in order to have the binding between the two strands. But now, if what's happened if we modify these two strands with the antigen? Okay, in this case, what we will have that the antibody will bring the two strands in close proximity. So thanks to the effect of the colocalization, this binding curve is shifted toward left. So why this is important? Because that means that we can use this strategy to detect the antibodies. And this is what we have done. The difficult, the tricky stuff in this kind of design is to find an affinity that is to be low between the two strands, but not so low, because otherwise we don't have the binding also in the presence of the target. So we tested, for example, in this case, 4 different variants of this strand. The difference between this strand is the number of bases that are complementary to the DNA strand that is attached on the surface. And how you can see here, I don't use the terms DNA output strand capture, but module. Why? Because in this case, in order to have the modularity, like in the previous example, we design a second strand of DNA that is modified with the antigen. Because in this way, we can just changing this strand, this second strand, in order to allow the detection of different kind of target. So the first step was we started with a DNP, that in general, DNP or DIG are used for proof of principle concept because they are cheap, and also because they are possible to buy from different company. Directly the DNA is modified with this kind of small molecule. And the first step was to performing binding curve by using different strand of this strand, okay, with a different component in bases that are complementary to the capture module. And how can we see? We will have that when we have just 6 bases, okay, the affinity is too low. So also in the presence of the target, we don't have a good signal. And changing the number of bases, we will have that increasing this the number, we will have that the binding curve shifted toward left, but decreasing the signal, because also in the absence of the target, we will have a higher background. So we selected the 10 bases in order to move in the second part of the work, in which we want to test also other different targets. And here is the binding curve that we can obtain by adding increasing concentration of anti-DMP. And how can you see we obtain a very good result in terms of sensitivity. And also the most important thing is the time of this, the response time of this kind of sensor that is quite fast. And also the sensitivity is really, the specificity is really good. Obviously, in this kind of application, what is really give the specificity between the antigens and the antibodies. So it's really dependent on this couple, not obviously of the DNA. And this work also in serum. And what we demonstrated is that by changing the recognition element, we are able, with the same technique, to recognize the anti-dig antibody by using the P17. We are able to detect the anti-HIV antibody. This is really important, is that the H19 that is is recognized by the trastuzumab, that is the most widely used antibody in the immunotherapy for breast cancer. And okay, and this is the DNP that is what we have seen before. Moving to the aptamer, I don't want to spend many words because Claudio already did. And it was just to say that, okay, one way to detect targets that are not oligonucleotides, that are not DNA or RNA, we can use or the DNA-like template or aptamer that, like Claudio showed before. Okay, this was the reason why I use aptamer in comparison to antibody. I just want to say that from my point of view, the most important thing is the synthesis of the aptamer that allow us to have a really reproducible result. Because in comparison to the antibody, we don't have the difference that we can have batch to batch that are due to the way in which antibodies are obtained. Okay, Claudio show you the very nice a lucky case in which we have a really good conformational change. So that means that when we attach the DNA, the aptamer on the surface, we can have a conformational change that brings to a different electrochemical output. But this is not true for all the kind of aptamer. And also because they are really dependent, like Claudio told you, that is depending on the way in which the selection of the aptamer has been done. So in many cases, the aptamer work in the meaning that they present affinity, but we cannot see when we immobilize on the electrode surface or when we modify with the fluorophore quencher, for example. But when we do, for example, a gel electrophoresis, we can see the binding. So what's happened in this case? That clearly means that the conformational change doesn't exist or is not enough in order to see for something that could be due to the instrument limitation, for example, for the concentration that we use. So in many cases, we don't have this conformation that allow us to have a really nice binding cut. So different strategy can be used in order to have a conformational change, to have a structure switching mechanism. One of these is that is called aptamer beacon. In this case, what we can do is to start from the native aptamer and modify. Oops. Sorry. And modify with some bases to one extremity that are complementary to a portion of the native aptamer in order to have this stem loop structure. Because we imagine that when we have the target will bind the aptamer, we will have a conformational change. Okay, in this case, if we modify, for example, in the easiest way with a fluorophore and a quencher, we will have that in the absence of the target, we will have a low fluorescence signal because we have that all the fluorescence is quenched by the quencher. In the presence of the target, we will have the conformational change. The fluorophore will move far away from the quencher, and we will have a high fluorescence signal. Is it easy to do this? In theory, yes. In practical, not so much. In fact, this is the first work where this strategy was proposed in, and they used the thrombin aptamer. The thrombin aptamer is the aptamer that is widely used, exploited, because it has a conformational change that is really impressive. Okay, but what they did, they this portion, okay, excluded the red one, is the original aptamer, okay, and they added these bases, okay, that are complementary to the aptamer. In the presence of the thrombin, we will have the conformational change. So from the low fluorescence, we will have a high fluorescence signal. But what is important of this work? That we can see what is not really easy, because in this case, they tested 3 different variants, and the difference of this variant is the number of bases that are complementary. And they used 4 bases, 5 bases, and 6 bases. Okay, when they have 4 bases, they have a decrease of the signal. When they have 6 bases, they have no variation of the signal because this stem is too strong, and so the conformational change is not allowed. When they use the 5 bases, they have a really good signal change. And so this is mean that in the most of the cases we have to do a trial and error, because also if we try to use software for, like for example NUPAC or other one, we cannot discriminate one basis, because the difference between these three variants is just 3 bases that are also consecutive. So that means that in many cases the work to do is not so quick. Another way in order to create a conformational change when we use aptamer is that it is called duplex aptamer. In this case, we have to design a strand of DNA that is complementary to a portion of the aptamer, and what we expect also in this case, and then when the ligand, the target, bind the aptamer, we will have a conformational change and the release of the DNA strand that we designed. And this could be exploited with a different kind of application for inflorescence, optical, electrochemical, but just modifying this strand. For example, in this case, that is one of the first work in which this strategy was proposed, we have still again the thrombin aptamer that was modified with this tail in order to attach these two complementary strands, one modified with the fluorophore and the other one with the quencher. And what we have that in the presence of the target, we will have the conformational change of the aptamer, okay, and the release of this strand. So in this case, we have the strand with the fluorophore will stay here, and this with the quencher will move away, and we will have a high fluorescence signal. So these are the results, in fact, where we can see the increase of the fluorescence in the presence of the target, also the binding curve. The last strategy that also can be exploited is that of the called splitting aptamer. In this case, we have to split the aptamer in 2 portions. These 2 portions can upsem just in the presence of the target. So also in this case, by modifying the 2 strands with with optical tag or a different kind of tag, we can have a different signal. For example, in this case, 2 different population of quantum dots are modified with the 2, each one with the one of the 2 splitted aptamer, and what we have that in the absence of the target, we have the quantum dots are dispersed in solution, and we have this color. When we have the target bring to the self-assembling of the quantum dots, and so a difference in the peak and in the color. Also here you can see the different size of the quantum dots in the absence and the presence of the target, and also the PM images. Also in this case was used the thrombin aptamer like proof of principle, so cutting the aptamer into 2 portions, and we can see when we have the target, we have the assembling, and so we can have the aggregation, and so the decrease of the fluorescence, and also the binding curve that is present really good result. Moving to the real, more or less real application is how can we use this strategy for improving also the sensitivity of our sensor. I mean, the contrary, I want to say the contrary. Okay, so another way in order to increase the sensitivity is to focus also on the transductor. That, for example, in this case, we are using in this work inject printed electrode that present a really high nanostructured surface. Why this? Because one of the difficult step is when we move from proof of principle to real application, because all this work the most are really nice, really fancy the most, but many times if you have a transductor mechanism that could be relied on a fluorimeter or a rod electrode, then many times the sensitivity is not enough. And one of the ways is to work on the, for example, on the porosity or the electrode itself. And in this way, in this work, we use this kind of electrode that we produce in the lab that are inject printed electrode in order to detect the N gene or 1AB gene of COVID-2, and for a multiplex detection. In which way? In this case, we use two different redox tag, methylene blue and ferrocene, and we design a sandwich strategy. So this means that we detected our target, the engine F1, by designing two different strands. One that is called the capture strand that is attached to the surface, and the second one that is the labeling strand that is modified with methylene blue, for example, for the detection of ORF1AB, and with the ferrocene in order to detect in the same time the engine. And we tried to move in more in a real application by designing also an antenna in order to do this kind of measurement directly by using the smartphone. So this is what I want to say, that we design this kind, we produce this kind of electrode that have a high porosity, and this is good because allow to load a higher concentration of a DNA strand. So when we work in higher concentration of probe, in general, we can decrease we can increase the sensitivity. In fact, this is the comparison between the active area of our electrode and a screen printed electrode, both made with gold. And we also were able to test two different probes. The methylene blue and ferrocene that we used, we can see, like you can see here. And this is the real device with the app that we developed in order to do the measurement. And these are the results. So we were able to detect in the same time the ORF1AB and N gene, also with a really fast kinetic. I don't know if I have time. Okay. Yes, okay. Because another, I just want to give some spot, okay, on CRISPR-Cas technology, because in the last year is one of the most, in many cases, really useful in order to increase the sensitivity. Because the CRISPR-Cas work in order to generate sensor, we can use the same, more or less the same strategy, but by modifying a little bit in order to use CRISPR-Cas. Because CRISPR-Cas work like a catalytic reaction. And briefly, okay, CRISPR-Cas is composed by an enzyme, the Cas12a, and the RNA guide. Okay, when we have this complex can recognize a specific DNA sequence that have to contain this that is called PAM motif. Okay, when we have this PAM motif, the CRISPR-Cas enzyme bind the DNA, the duplex, okay, and start its activity. That we have the cis activity that work on the duplex, okay, cutting the duplex, our target, and this give not a signal, obviously, but this is used, for example, for editing. But how is really useful this kind of system for biosensing, because we don't have just the cis activity, but when this happens, we will also the activation of that is called transactivity. That means that we have a nuclease activity that allow to cut, to cleave all the DNA that are in solution. So, for example, if we have in solution a DNA strand is modified with the fluorophore quencher, after that we have the activation of the CRISPR-Cas enzyme that we have just in the presence of the target, we will have also the increase of the signal because it's due to the transactivity. Why this is important? Because in this case, we are not limited by the concentration of the target, because what gives us the signal is the reporter. The reporter that could be in a really high concentration. So that means that we can expect a really high value of in general fluorescence, because in principle this strategy was used in fluorescence because it's the in general. I worked on both technique, electrochemistry, and fluorescence. For sure, working in fluorescence is easiest that in electrochemistry, also faster when we need to verify a theory. Okay, and in this case, for example, when we have in solution our target, we will have the cleavage of the target and then also the cleavage of our reporter. Reporter that in general are a short DNA sequence with the fluorophore pair to the extremity, and that allow an increase of the fluorescence. This method is a Collet detector. It is based on the use of Cas12 and an RNA guide, and are able to detect and cut cleavage DNA sequence. The same, the real is the same principle, more or less, is that is used in this other technology that is based on the use of Cas13. In this case, the target is an RNA strand, but the mechanism is the same. So we have an RNA guide that is designed in order to recognize our target, and then we will have the cleavage of the reporter and the increase of the fluorescence. And this is one is been many work was done have been done during the COVID, because, for example, we are able with this technique to recognize the gene of COVID with both by recognize the RNA or the DNA. In general, I just want to give an highlight on this work that it was the last that I did when I was in Tor Vergata, that was on the study of the reporter, because in the many of the work that you can find, the reporter that was using was a really short DNA sequence, in general 6 or 6 timing, modified with a fluorophore quencher. What we can try was to try to move from the linear single strand to DNA to a stem loop reporter, the same molecular beacon that I mentioned at the beginning. And what we discovered that by using a molecular beacon, we increased a lot the sensitivity. In fact, like you can see here, we designed also in this case different variant. The different variant are different for the length of the loop, and we discovered by increasing the loop with 10, for example, with the 10 timing, we can have a big increase in the fold change, because for this is the result that we can obtain, okay, with the normal one, and then we can have that by increasing the loop, we have a big increase. And so we move then when we arrived in the Mercosi group to use this finding in order to create a sensor for the detection of, in this case, of bacteria, Escherichia coli and Staphylococcus aureum. By in this case, what we have done, we designed the RNA guide, because the thing that we have to do when we want to use CRISPR-Cas is not really complicated, to be honest, because it's just to find a PAM motif in the target that we want to detect, and then to design the guide, because the guide is fixed. There is a portion that is fixed, and the other one that we have to design that is complementary to the target. Okay. So what we have done is to design 2 RNA guide, one for the Escherichia coli, the other for the Staphylococcus aureus, and then to attach the molecular beacon that we studied in fluorescence in electrochemistry by modifying this molecular beacon again with the thiol group to one extremity and methylene blue on the other one. And we can see that we were able to detect both the bacteria by just changing the guide. And these are the results that we obtain in real sample. when we can see that by using the guide for Escherichia coli, we are able to detect the sample with Escherichia coli. This is the sample that contain the Staphylococcus, and we don't have the big signal gain. In the other case, by using the guide for Staphylococcus, we can see just the peak increase for the sample that it was positive to Staphylococcus. And that's it. And in the last, I just want to show you briefly the main project in which I am involved now. That is about the detection of Alzheimer, more the detection to see, to monitor and the Alzheimer disease. In this case, we want to move from a simple electrode to electrochemical lateral flow in order to combine the advantages of both. Danny, for sure, will explain better than me all the advantages of lateral flow. In this case, we want to combine the advantages of lateral flow with that of the electrochemistry. And in this case, by using aptamer like a recognition element. And the idea is to detect different biomarker by the use of aptamer that can have a conformational change. Conformational change that is detected by using electrodes that are directly printed on the lateral flow. So my conclusion is that I try to explain a little bit how can we use DNA nanotechnology by using different kind of technique, and by using DNA not just only for the detection of DNA or RNA sequence, but also for a large wide of analyte, like it could be a small molecule protein, by using aptamers, or also to use using DNA like a template. So that's it. This is my acknowledgement to all the supervisors that I had, and in particular the group of Torre Vergata, where I spent many time, and the current group. Okay, that's it. Thank you.
Okay, thank you, Maria. We have time for one or two short questions. Okay. Oh, sorry, take a microphone.
We didn't use any kind of chemistry. You just modified them with style, and that was enough for attachment of the gold. Okay. Okay. Okay.
Okay. Next time, another country for the workshop. Okay. Okay. In the case of gold, the most used chemistry is that of the tile with the gold. This is really well exploited, and okay. And then Andrew will give you more details on this. And but yes, it depends on the material that you have for the functionalization. For example, if you work with 2D material, like for example graphene oxide, use graphene oxide, you can exploit different kind of chemistry. But in general, it's not a complicated kind of chemistry, or it's a chemistry, but are basic reaction. But yes, of course, the most important thing for, in my opinion, but in general, many times could be used, for example, just absorption when you work in electrochemistry, independently by the bioreceptor. But in the case of DNA, if you don't have a good self-assembled monolayer, you cannot reach a good result.
Yeah.
Because it's, for example, or you don't have the conformational change, or I mean, it's really important in the case of aptamer, for example, to have a really good self-assembled monolayer.
Mm-hmm. Okay. Okay. And also for optimization of parameters, did you use any kind of statistical tool or RSM, or you just chose the different concentrations by just simple choice? I don't know. By
Okay. Okay, in my experience, is the most are trial and error, but also because more or less we know many stuff. I mean, meaning that, for example, if we want to test, for example, the concentration of magnesium, for example, that could be useful or not, depending on what we want. Okay, if we want to stabilize or not, for example, the DNA, we know that, for example, the tests that you can do are quite limited because, for example, the maximum concentration that you can use is 10 millimolar. If you do a test without magnesium or 3, 2 millimolar, 5 and 10, you have already an idea. But for sure, to extend the experimental section also to artificial intelligence, other stuff, obviously could be really useful. But in my case, for the most, is just question of error and trial. But
Okay.
also having in the group many competence in this. So we already know in which way we can move.
Okay, thank you. And the presentation was great, and also your research. I forgot to tell that.
Thank you.
Thank you.
Hello, thank you for a really nice presentation. I wanted to ask you about inkjet printing of gold nanoparticles. So did you make this ink by yourself or use some commercial ink?
No, we didn't do we print the electrode. I'm not an expert in this, okay? But I don't know the company that produce this kind of ink. But what we done is to use this ink, and then we have to do a sintering process in order to have the conductivity. And yes, but we don't do the ink.
Okay, thank you. And just a comment. So I see that I read somewhere that it said people are moving away from this fluorescence quenching type of sensors, but I think it's still a good standard for, let's say, like you said, confirmation.
Yeah. Okay, it depends on the application because fluorescence could be useful in many cases. Fluorescence is the best because, for example, if we have to work with a cell, for example, obviously the fluorescence is still the best, but it's depending on the application. Because, for example, if we want to think to point of care, obviously electrochemical, but with screen printed is another way to do more faster. And also because the other important thing is the consume. I don't know if it's for the English or not, but because, for example, if we want to use the smartphone, the energy that we need for fluoroquencher is higher than if we have to use electrochemistry, because the energy power that we need is completely different. And this is the reason why there is already a balance between what is the final application.
Okay, thank you again.
Thank you to you.
Okay, so to be on the time, you can ask additional question during the next break. So
Okay.
thank you, Maria, once again.